Hello, I'm Meredith Blackwell, and this is Don Pfister, and we're here as part of the Oral History for Mycology project. We're on the campus of the University of Florida in Gainesville, and today is the 12th of July, 2022, in the Hilton Hotel on the campus. And we're here to interview Nhu Nguyen for our first of the morning. Don?
Thanks for coming out, Nhu. What we tend to do here is try to get some background about the people, where you're born and so forth. Ultimately, we want to know how you got to fungi. But so you can begin. You have a very rich background here.
Wow. Okay, so I was born in southern Vietnam, at the very tip of southern Vietnam in the Mekong Delta.
And what's it called? Is there a city?
Yeah, city is Châu Đốc, and it's very close to the Cambodian border, so you can literally walk a few miles and get to the Cambodian border. But I grew up in Delta life, which means lots of water and rice fields. I got exposed to a lot of nature when I was growing up. We have a small family farm. And so my parents were farmers, and we have a small orchard. I played a lot in these orchards. I didn't play with a lot of insects. I didn't get to play with a lot of fungi. I didn't know what those were back then.
Let me ask you one thing. You mentioned a lot of water and you were exposed, and I thought you were going to say to a disease. Was there malaria in your area?
Probably, but I don't know about it.
Remember.
I don't remember. I was pretty young. I moved to the States when I was about 9, into a different delta, the Mississippi River Delta, in southern Louisiana, and yeah.
And I've known you for a long time, so I know about something about your father. Do you mind talking about that a little bit in his orchids?
Yeah. So my father inherited a lot of land from my grandfather, and he grew a lot of plants. And my grandfather actually collected some orchids back in the 50s when the Communist Party took over the North. People fled south, and so they needed more land for people to establish. So my grandfather was part of a group to expand the land. So he was in the jungles, and he brought home orchids, and that's where we still have some of his orchids until my uncle's still taking care of them in Vietnam today. But so it got me fascinated with some of these really interesting plants, and I still like growing orchids.
And plants that were associated with fungi that you didn't know about.
Yes. Not until many years later, when I as a second semester freshman, I emailed some professors to see if
How many?
I think it was 3 or 4.
How many answered?
1.
Was I ever lucky? Was I ever lucky?
I have to be the lucky one here, because I had no idea I got a response from a famous mycologist.
So were you a biology major at that point?
I was a biology major. I wanted to, I dabbled in entomology, and I wanted to become an entomologist. And since Meredith's lab was working on yeast in beetle gut, that got me really excited. And yeah, so I joined the lab and got pulled into mycology in such a wonderful way.
Yeah. So tell us how it was working in Meredith's lab. We're going to grill her a little bit and see how things are. But this is firsthand.
It was for an 18-year-old who's really interested in science and wanted to be a scientist. It was incredible. I think I get there every morning, 7 or 8. Meredith would already be in her office, picking away at the keyboard, and we had a postdoc, Soy Su, who trained me in molecular biology. My first PCRs and DNA extractions.
And I should point out, he's the real teacher.
but it was great because you would be there, and Sue would be there, and there would be a few other students. But I got to learn how to work with fungi, and I learned to work with yeast. Got exposed to lots of things in mycology, and once in a while I get a call from a guy called Don Pfister, who is like, "Is Meredith there?" I say, "Yes, he's around somewhere."
Well, and the really good thing was you had the interest in insects, and that was one of the problems, finding someone that didn't mind dissecting insects.
So
he was good at that.
you went out in the field. You did field work with them.
Mm-hmm.
Talk about that a little bit.
So we were collecting. What were we collecting? Beetles in
the fungi and
fungi.
and also the phthalates.
And also this large beetle that lives in wood.
Mm-hmm.
So
Usually white rotted.
White rotted wood, and we would do a lot of find a mushroom or some kind of a crust fungus, collect as many beetles as we can before they fly off. And for the wood-feeding fungi, we hack off logs and try to find these beetles. There'll be few dangers, chiggers and other.
I was always worried someone would be bitten by a snake.
Yeah.
Because they're right under the logs.
So you collect the beetles, then you must have processed them too.
Yeah, we collected beetles, bring them back to the lab, dissect them. The least fun ones were seeds, which are about 1 millimeter long. Those are not as much fun to dissect, but I got good at dissecting these tiny things.
And where did you collect?
Everywhere, right? We mostly Louisiana, but then I got a chance to. You took me to Panama, to Barro Colorado Island, which is incredible. First experience in the neotropics, and that's fantastic. So we collected there, and we got some cool stuff out of those beetles.
And one time in Georgia.
That's right, one time in Georgia. And you sent me here to Gainesville to collect a second rare species of psalid.
Yeah, talk about that collection. That collecting trip.
That was a fun one in some ways. I flew into Gainesville on the second to last flight before a hurricane hit. I managed to just squeeze through, spend the night here at my friend's place, and then the next day we drove down to Archbold Biological Station, which is a wonderful place. Really lots of interesting things, and we spent our days hacking at the stumps, the dead stumps of oak trees, because this beetle lives in these dead stumps. Hacked for a whole day and a half, and didn't find anything.
And meanwhile, the hurricane was coming in.
The hurricanes already came in the night before.
Okay,
It was
it was over.
It was a quick hurricane. It was over.
A lot of rain, though, still.
A lot of rain. We saw a lot of high wind that bent all the poles, and yeah, it was a decent sized hurricane.
Uh-huh.
But
Did you find it?
unfortunately, we didn't find it at the time I was here. But as soon as I returned home to Baton Rouge, the entomologist called and said, "Hey, we found this thing walking across the street, so we'll send it."
Yeah, we actually, I think, had a total of 3 isolates, or 3 beetles, I shouldn't call them isolates.
Yeah.
But they were interesting because they had a different different ecology, and in the 3 that we had, which is a very low sample, we never found the xylosperm yeast
Wow.
in them.
So you finished up at Louisiana.
I did, yes. I finished up at Louisiana.
And you stayed an extra year.
I stayed an extra semester, I believe.
Why was that? Learn more about insects.
I actually can't remember.
That was the reason you gave me. You wanted to learn more about insects, and I was afraid you were going to become an entomologist.
No, actually, I finished my undergrad in 4.5 years.
Okay.
That's what it was. Because I actually spent all my summers working in the lab, and I didn't care about taking extra classes because doing research was so much fun. So I finished in 4.5 years, so I have an extra semester before graduate school. So I spent that extra semester.
And I should mention, I usually didn't take freshmen in the lab because the next year they would have to have organic chemistry. But it didn't matter with Nhu, his first organic, you made 100. You remember that? He made hundreds on your organic chemistry test, which was something. And
Yeah.
although he spent all his time in the lab. But he's not through with the story. You got papers out, and they were first authored papers.
Yes, my first paper, well, the first paper I wasn't first author, but that was in 2003, I believe. It was also on a yeast beetle associate. And then I got a couple of papers out before I got to graduate school.
4, I think.
We described a number of interesting things, including one that's really well cited, Spathaspora.
A new genus he named.
genus. And that's being used now for all kinds of biofuels work.
Really good fermenter of xylose.
Good.
Okay, now we can go.
Now, you're 4.5 years, 4 or 5 papers. And what's next?
Graduate school, that's the obvious choice. I applied to several schools, and I narrowed down to Wisconsin at Madison and UC Berkeley. Meredith didn't say very into my face, but I can tell that she wanted me to go to Berkeley. Because at Berkeley there was actually a good mycologist, Tom Bruns,
Yeah.
who pioneered molecular ecology for fungi. So that was a hard choice. Actually, I liked Madison. I liked its program, but I wanted also to experience something new and different. So I went.
So what Meredith said didn't
No,
say
what I
Meredith
didn't
went.
say.
It happened.
No, I just thought they would be a good fit, he and Tom.
And obviously it was a good fit, huh?
It was.
I thought the informalness, you were used to that and doing as you please, and I thought you might be able to do that.
Well, doing as I pleased. I did get to do a lot. I pitched some ideas, and you and Sue would always say, "Yeah, sure, do it." So Tom would also be, and is that way. So I got to do what I wanted in Tom's lab, which is to study bacterial-fungal interactions. Not in those same terms back then, because I didn't know what I was doing. But eventually, that's what it became. Yeah.
You also did a paper on a discomycete.
So yes, at Berkeley, I made friends with Elsa Wallinger, who's a great mushroom taxonomist, and we did all kinds of weird fun things, starting with some Sewell's taxonomy, and we said, there's this thing called Helvella, and it seems to be really common, but what's going on with it? Is it really common? As it turns out, we try to find specimens, and there was no specimens to be found, at least in herbaria, because nobody was collecting it because it was deemed so common. So it actually took us a little effort to get the common specimens, and we eventually published two species. That is sort of the West Coast variant of the Helvella lacunosa complex. So that was fun.
Good, yeah.
And another paper on onions.
Are you trying to tell me that I'm everywhere?
Yeah, no. I think it was pretty nice that you were able to do rotations and get something out of each rotation.
I think you told me that. You're the one who says, you really need to get a paper out of your rotations. So I did. I went to these rotations to the professors, and I said, I'd like to get a paper out of whatever I'm doing. So I did get with Chelsea Specht, which is a really fun lab. And I rotated there just for fun because I really liked the way that she operated, and did a paper on the Western onions, the Western California onions. She wanted me to join her lab, but the pool of mycology was so strong.
Couldn't let it go. Yeah.
And then you got another one, and you got me involved with the people at the Lawrence Berkeley lab, which paid off for us because they gave us money. They were interested in his xylose fermenting fungi.
And we did a little bit of a paper. Back then we had phylochip, which is this great new technology to characterize bacteria using environmental DNA. So that was pre 454, pre Illumina sequencing. So we characterized the microbes in bacillus beetle guts using these phylochips. And that paid off too. Eventually, we got a paper out of it. Yeah. So that ended my rotation, which actually became my first chapter in my dissertation.
And so then
You finished up there.
Yeah, I finished up at Berkeley, and went to the University of Minnesota. Now, this is when Peter Kennedy just moved from Lewis and Clark to Minnesota.
and you had known him at Berkeley?
I had overlapped with Peter for just about a month, I think. He was finishing up his postdoc there in Tom's lab, and went off to Lewis and Clark. So we sort of chatted over the years for a little bit because I had an experiment that I was running and I needed some help. Yeah, when I was looking for a postdoc, Peter just happened to have arrived at Minnesota, and so I joined him in Minnesota in 2013.
Yeah.
Okay.
Fall 2013.
We interviewed Peter yesterday, and he talked about your influence in the lab and your help in the setup.
You did a lot of ordering.
I did a lot of ordering. I set up the lab, and I have my particular ways of wanting to do things, and Peter let me do it. so I appreciated that. And I basically set up the lab to be able to do high throughput sequencing on fungi. So that's my contribution with Peter.
How long were you there?
I was there for 2 years. I wrote with Peter and Meredith a grant to the NSF to study Suillus. We submitted it right before I left, and a couple of months later, Peter called me as I went back to Berkeley, and Peter says, "Nhu, you remember that Suillus grant we put in? It got funded."
Nice way to start out.
But by then, I've already accepted a position with Mary Firestone at Berkeley to do a second postdoc. I wanted to learn about stable isotopes and really get a little deeper into bacteria interactions with fungi. So that's where I ended up for, unfortunately, just 9 months before I accepted a position at the University of Hawaii at Manoa.
And that's on which island?
And that's on Oahu in Honolulu.
It's the main campus.
It's the main campus, yeah. Yeah.
So you get to Hawaii. You are teaching. What's your activity there?
Yeah, so I do mostly research. My appointment is mostly research with some teaching. So I teach.
And you're not in the biology department.
I'm not in the biology.
What department are you in?
I'm in tropical plants and soil sciences in the agricultural college. So my responsibilities for teaching is soil biology and a graduate level class on soil microbiology, broad ecological type things. And I get to finally, after a few years, design a mycology course with Nicole Hinson and Anthony Amon. And we three teach mycology course in spring. So that's been fun.
And have you risen through the ranks very far?
I did get tenure. I was awarded tenure last year. Yeah, so continue to pull forward and yeah.
Talk a little bit about fungi in Hawaii.
Well, that's
It's a big topic, I know, and there have been a number of mycologists. I mean, we're talking about history of mycology here. Gladys Baker was in Hawaii, Roger Goose, Don Hemmes. There are probably more that I've missed.
Well, and then Don started having lots of mycologists go from the U.S. to
Yeah.
collect. Bob Gilbertson.
Yes, and their footprints can be found in the specimens.
That's good, because I was wondering about that, because some people don't know about Carling that are at Purdue, and so there seems to be a lot of forgetting who started in the past.
Yeah.
But the footprints are there.
They're in the Bishop Museum. So a lot of the specimens were deposited in the Bishop Museum, and recently I tried to pull all the records of macrofungi from the museum and other places to try and get an inventory of what's there. And there were groups that had names. Xylarias had names. So
Sure.
lots of wood feeders, and so you can kind of trace the history back a little bit. Mm-hmm.
Jack
Yeah.
Rogers.
Jack
yeah.
Rogers.
Jack
It
was
was-
one of the ones. I should have named him.
Yeah, that he went. I know that, for example, Roger Goose, I think many of his sooty mold work that he did in Hawaii, the black mildews, I think that was something that he did when he was there. And do you know much about Gladys Baker? Do you know?
I know a little bit, but not a whole lot.
Yeah, she was very interesting. She trained as a lichenologist, and she worked with C.W. Dodge and St. Louis, and was part of the papers that first addressed the Antarctic fungi. And among her first publications were those lichens from Antarctica. And then for years she was at where? Poughkeepsie, New York, with the college there, the women's college in Poughkeepsie. But she then went to Hawaii and was kind of the contact for people in Hawaii for years.
Yeah, and then she moved to Sun City, Arizona.
Yeah, that was in
She
retirement.
retired, yeah. But the interesting thing, she had some very dedicated students, I guess, or associates, because I think it was June Long at Syracuse who started this. There's an award named in her name, and Baker. Wait, it's who does she work with? It's Baker something else. Martin Baker. It is.
Martin Baker.
Yeah, and that's because of June. June, I think, worked with Martin too.
Yeah.
So I don't know if you've ever applied for that when you were a student, Martin Baker Award.
I don't think I did because I didn't qualify for that area.
What the qualifications are.
Yeah.
But she's remembered there, and we used to give out a biography of all of the people that were named in the award, that the awards were named for, so that the students would know who they were.
Yeah.
I don't know if that's still done or not.
Yeah.
And you have then Don Hemmes came in.
Yeah.
Did you interview Don?
I don't believe we have.
No, we haven't.
Yeah.
Yeah, he's here, and
Yeah.
Helen, his wife, and
Yeah.
and Don has a sideline. I don't know if everybody knows about it, but he's described a number of little mollusks, very, very tiny ones. He's a real mollusk collector. And you've got some sidelines, some hobbies.
Yes, I have described some recently described a tiny little fly with Mike Farrow at Clemson. It's an extremely tiny fly.
And that was an LSU connection you had made.
That's right.
Mike.
Yeah.
Okay, and then also outside of mycology, he's a great cook. He also is a great painter. Are you still painting?
Not as much as I'd like to, but I'd really like to get back to it.
Now that you have tenure, and you probably got some support, right, from grant money, and
Yes.
yeah, you've been very active doing that. So
I got some good support from various agencies, so I'm happy about that.
yeah.
Yeah.
And what are those supporting? What kind of work are your grants supporting?
So I have some of the main lines are in plant microbes interactions, mostly in the root system, because technically I'm supposed to be a soil microbiologist now. but those kind of support plant interactions come from the USDA, and I have some carbon work, trying to really put fungi in the forefront of climate change. We know that fungi is important, but it's really hard to get people to understand that, even people who study microbes. So that's part of the work, and part of what I'm trying to do is spread the word that fungi is important, and we do that through our science.
And how are you presenting that? How are you getting it out into the world?
So far, not very far yet, because because I just got a grant to do such.
And what are your plans then?
Well, my plans would be do the good science, present it there, and then we will try to, if we find something that is relevant and notable, we'll spread it through various media. I do have Twitter. I don't use it very much for anything else other than academics, but it's a great way to also get papers.
Yeah. And you have certainly done work before while you were at Hawaii until now, other work,
Yeah.
because you did get tenure and you
Yes.
have a number of publications.
The other work is mostly on the USDA side, because my college is an applied college, so I do have some applied work with plant, fungi, bacteria interactions. But now I'm trying to move a little bit forward with fungi and global carbon cycles.
Uh-huh. Good.
So in your 9 months with Mary Firestone, are you applying any of what you learned in Hawaii?
Not quite, because it was short. Mary just started on this project on switchgrass, and what I really wanted to learn was how to use stable isotope. And so I didn't get to do that because the project was just starting, but I managed to keep a connection with Mary and be on the next project that she had. So now I'm starting to pick up those skills, which I think are going to be really important, and they're really fun to work with fungi, because fungi can transport, and you can use that technique to trace where the carbon is moving. Yeah.
Anything more you want to say?
Well, I appreciate the mycology societies, mycological societies' support over these years. You can't find a better society that supports students, and in your support, pushing me to go do things, but do fungi.
Yeah, he went and did things in Panama, like they had a reenactment of a photograph from 1950. He and Mike Farrow and another entomology student reenacted this photograph. They had beetles. What were you doing with the aerotilids? Were they fighting off?
Yeah, we were just poking around the forest, first time in the neotropics, and you see all kinds of things. You see army ants that you've never seen before. So we were throwing these beetles into the ants' path, and some of these beetles do produce chemicals. The ants just kind of walked around them. Pretty cool stuff.
I'd never had any idea the beetles did this.
Yeah. So just exploring fun things.
Yeah, no.
It was fun.
Well, that sounds great. That's what often we like to do.
Yep.
And
Yep.
you've taken advantage of it quite well. Yeah.
So thanks for talking to us.
Thank you, and I was I'll be with fungi forever.
Yep. Well, that's good. Thank
yeah,
you.
bacteria and fungi is big now. So
But
that's good.
bacteria are a symbiont of fungi.
Yeah.
Yes, fungi are still that thing.
Okay.
Yeah.
Okay.
Good.
Thank you.
Thanks.
Thank you very much.